And we're now in a programme that is a multi-collaborative BBSRC funding project with a multitude of different authors led by Ian Toth at the James Hutton Institute. What it's looking at is the mechanism and the control strategies for making a decision support tool in Blackleg. And obviously Blackleg is very heavily implicated with the conditions in the field, like temperature and moisture and the presence of the bacterium. So what we're looking at here is the control strategies for managing irrigation with crops that are susceptible to Blackleg. So we're using here different levels of inoculum loading on a variety called jelly. So we've got low, we've got high, and we've also got some control mini tubers where ostensibly there should be no disease whatsoever. What we're superimposing on that is a combination of different irrigation timings and amounts to either induce the disease or prevent it by managing it. So this is a 3-year program. It was subject to COVID variation right at the beginning when the project's start was really threatened by all the things that happened in March and April. We managed to plant, albeit quite late in the early planting program, but we've managed to get on and we've currently applied 2 irrigations to this crop and we're well on the way to doing our experimental program. So we're following this in terms of development of the disease, in terms of symptoms, then looking for rots at the end of the season. In collaboration with other partners at JHI, basically looking at free-living nematodes, we are actually doing some work off-site to sample the soil and look at how nematicide interacts with free-living nematodes in actually preventing the invasion of Pectobacterium into plant roots. So that's a key part of the program, is this collaborative approach. And we're also working with the University of Glasgow to look at monitoring soil for the assays involved in Pectobacterium in the soil. So it's a start of a 3-year program. We've got it underway and it's progressing nicely.